you what, it has just been a remarkable day already for us in our world. But good afternoon to everybody, and thank you for joining us this afternoon. One of the major themes that emerged from our recent conference and our global regulatory and scientific forum was the future of therapeutic development depends on collaborations. And not only among the researchers, but among the patients, the clinicians, the advocacy organizations, and countries working together for the common goal of finding that therapy for us, right? Today is a wonderful example of that collaboration in action. Our discussion will focus on developments that have taken place in Canada and how the Canadian patients can play an increasingly important role in advancing research alongside every one of us. We have the pleasure today to introduce ZIV GAN-OR, an associate professor of neuro, and he is working in concert with our SP CERN and Dr. Fakari and our folks in Brazil. And we have this amazing global conference that I want to make sure that you guys get a chance to see on the YouTube channel. So grab that link and watch that day, and you're going to get a chance to see some incredible discussions about HSP and PLS. But I want to take a moment to introduce Dr. Ziv, and then I'm going to bring it back around and let Linda LaFontaine, one of our very own HSP fellow comrades, ambassadors for Canada, she's going to talk about the cohort and what she has been doing, working with bringing all of those patients in Canada in line with the things that we are also working on collectively. So, Dr. Ziv, why don't you take over and we will listen to your presentation today, sir.
Thank you very much. I'll share my screen and I hope everybody can see it. So my name is ZIV GAN-OR. I'm a geneticist, or what we call the neurogeneticist, specializing in neurological disorders and genetics. And I'm located at the Montreal Neurological Institute, which we call the Neuro in short. It's a combination of a neurological hospital and neuroscience research institute under one roof. And I'm serving in this hospital. I'm serving as the executive director of the clinical research unit, which is the unit that runs all the clinical trials here. So we run over 100 clinical trials in parallel across basically all adult neurological indications, common diseases, rare diseases, etc. I'm recently appointed as the director of translational clinical research at the institute, which is I basically oversee all the preclinical translational research at the neuro, all research that is aimed towards developing treatments for neurological disorders. And I also have my own lab, which is a lab that is using genetics in order to understand how genes affect risk for disease, how genes affect how disease progresses, and how it responds to medications, with the goal of identifying targets for therapeutic development. I work with multiple pharmaceutical companies, and when we do identify targets and we have some functional validation, meaning that we test them in different models and we see that they are potentially good candidates to move forward in order towards development of treatment, we work with either with other research institutes and with pharmaceutical companies to do that. And today I'm here to tell you about our efforts, of course, on HSP, which is what you're interested in. I'm one of the co-leaders of what we call CAN-HSP, the Canadian HSP Network. So I will tell you a little bit about it in general. I'll give you an overview of how we structure, what we're doing, what we have been doing. And then I will end up with two projects that are more therapeutic in nature or towards therapeutic development. And I hope you enjoy it and ask me any questions that you'd like to ask, and I'll try to address all of them at the end. So CAN-HSP. So CAN-HSP is a Canada-wide registry basically of HSP started by the group of 9 people that you can see here. You see, I'm not one of them. I joined only in 2016. The CAN HSP started in 2012. By the top 6 are all clinicians that seeing neurologists, seeing HSP patients, and 3 at the bottom, they are basic researchers also doing, one of them retired, but the others are doing basic research on HSP and other diseases. And it started in 2012. We got funding for 5 years. So funding ran out a long time ago, but we kept running on, don't tell this to anyone, but we're using, leftovers from funding that we get from different sources to keep this going. I'm sure you know, I mean, you're a foundation that provides funding also, but it's very difficult to get funding. The success of SP CERN, recent success of SP CERN to get significant funding is incredible, and I think it will push the field forward towards finally getting some treatments for HSP. So CAN HSP, when it was established, it had 3 aims. There's or 3 arms, if you'd like. One arm was focused on clinical aspects, another arm was focused on genetics, and the last arm was focused on functional, meaning understanding the basic mechanism, the function of the genes, the function of the proteins involved in HSP. Clinical was more about how the disease progresses, the differences between different types of HSP, etc. So you can see the aims were not there's nothing therapeutic in it really. But since then we have shifted a lot towards that, and that's again what I will talk about more towards the end of my presentation. So the way CAN HSP has been working, through the clinicians that see HSP patients across Canada, they would recruit patients and provide their clinical data into our centralized database. For some of the patients, we would do panel sequencing, or the clinician would do panel sequencing. That was a long time ago when they started. That was the main genetic tests that were done. But today we're going directly into either whole exome or whole genome sequencing. We have a total of 426 families in CAN HSP with 671 affected individuals. Out of those, to about 65% of them, we have a genetic diagnosis. So we know what the genetic cause of HSP, meaning the 35% we still don't know. So there's a lot of research to be done in that aspect as well. So this slide provides a general overview on the population. So you can see the average age at onset is 15 years, but we have a very wide range of age at onset that I'm sure all of you are aware of. Some people get it very early childhood, some people very late at life. And you can see the graph on the right, how many of the cases are autosomal dominant, how many are autosomal recessive. I assume that you know this terminology, but if not, I'm happy to explain later if if I'm using terminology that someone doesn't understand, feel free to ask. A small portion is due to mutation on the X chromosome, and there's a large portion, about 1/3 of the patients, that is we don't know the mode of inheritance. Usually it's because it's one individual per family. So it could be a de novo mutation, or it could be a recessive disease that we just we don't know that it's recessive, etc. It's just unclear from looking at the family.
Mary,
It's just unclear from the family pedigree to know if it's the inheritance pattern. In terms of the genetics, our cohort, the Canadian cohort, behaves like most cohorts that you know around the world, in the sense that SPAS mutations, SPG4, are the most common. Then we have SPG7, ATL1, SPG11, SPG8. These are the top 5 most common genes in HSP in our cohort. And there's about 15% of the patients that we just have one patient with one gene. And as I said before, about 35% that are genetically diagnosed. And again, on the left, you can see other characteristics of the cohort that we have. So the sex distribution, the age of evaluation, the mode of inheritance that I already talked about. One thing that I want to mention is that when we've been doing all these genetic analysis, and I'm sure it's not new to many of you, to all of you, many of the people that are diagnosed with HSP can have other diseases that mimic HSP, so they present like HSP, but they're not. In our case, we found 70 individuals like that. It's a substantial amount. About 10% of the patients have mutations in genes that are not they're not the classical HSP genes, although the clinical presentation looks like HSP. I can give you an example that was very interesting. The second gene that you see on the list from the left, SLC2A1, we had 5 individuals from the same family. The family was diagnosed with HSP for over 2 decades. They thought they have HSP. And thanks to finally being able to do whole exome sequencing, we discovered that the mutation that they have is in this gene. It's causing a disease called GLUT1 deficiency syndrome that has spasticity as a major component of it, but it's not HSP. But the really interesting thing, or the really important thing, is that it's actually treatable. Not only it's treatable, the treatment is nutrition. It's ketogenic diet. So after a couple of decades of not knowing what they actually have, they now do know what they have, and using ketogenic diet, dramatically reducing their symptoms. So I just put here the list of all these genes in case someone is interested, but all of these are different disorders that can mimic HSP. And as I said, about 10% of our cohort are that, but we still include them in CAN HSP. So since we were established, we published as a group, as the network, 34 papers, and we contributed to the discovery of 12 genes, which is quite a substantial amount. 4 of those that are underscored here were discovered by ourselves. And I know maybe it's not that sounds that exciting, a discovery of a gene, but we think it's very important because it identifies a target. It helps the family in terms of genetic counseling, the ability to decide how they want to move forward in terms of the next generations. So those that are underscored were discovered by us. So I'm going to show you now a few examples of papers and talk very briefly about them, because I don't want to go too deeply into the science. It's more to give you an overview of what we're doing, but I will go a bit deeply into the science when I will talk about therapeutics later on. So these are one example on the left from 2012 and another example from 2016 of 2 important genes that we found to be involved in hereditary spastic paraplegia. And I will talk about calpain-1, which is also called SPG 76, sorry. I will talk about it later because we are starting a therapeutic program for this specific gene as well. Other studies that we've done, so on the top left, this is a more pharmacological study done on different models of SPG4 SPAST. So it was worm models, fly models, zebrafish models, identifying a potential. We did the screening of potential therapeutics and found one drug that we thought could be a good candidate for a repurposing kind of trial. But so far this hasn't moved forward due to lack of funding. And the other studies that you see, it's additional genes that we've discovered or we contributed to the discovery of. The reason I put this paper here, this is a very recent paper published about 2 months ago, is to show you that even when we think we know something about HSP genetics, we need to take it with a grain of salt, at least for some of the genes. So many of the genes we know for a fact they are HSP genes. This gene specifically that I put here, CPT1C, we had doubts about it from the very beginning, from when it was published. It was published from a couple of families where the authors found mutations in this gene, and until this day, there are some families that being reported in the literature as having CPT1C mutation. I'm curious to know if there's maybe somebody on the call who thinks they have a mutation in this gene. Basically, what we did is very simple. Now that we have access to sources like the UK Biobank, I don't know if you heard about it, but the UK Biobank is a biobank that includes more than 500,000 people from the UK that contributed samples, that all their medical records are inserted into the database, and it's a longitudinal study, meaning that every time somebody is diagnosed with a new thing, it's added into the database, etc. So we screened this huge database of more than 500,000 people, and we found over 170 people that have mutations, loss of function mutations in CPT1C. So these are mutations that supposedly should cause HSP, but exactly 0 of them had HSP. In fact, the most common thing that they had was people with these mutations, they had cataract and problem in their digestive system. So the conclusion of this paper of us is that CPT1C is not likely to be an HSP gene, although currently it is defined as one. It's SPG73, but we do not think it's true. And because of that, what we decided to do next, which we are now in the middle of, is to systematically explore all the HSP genes in the UK Biobank, but also there's the All of Us study from the US with also 500,000 people, and FinGen with hundreds of thousands of people. So different databases like that, where we're going to investigate all the HSP genes and see if there are additional genes that we do not have, that we have genetic evidence that it's actually not involved in HSP. So we are basically going back and validating previous studies. I think this is the last study that I'm going to mention from our group, also very recent. The reason I'm mentioning it because it's a very new concept, what we call, I mean, the concept is not new, but the discovery in HSP is new, what we call digenic inheritance. Digenic inheritance meaning that instead of having mutation in one gene that causes a disease, let's say SPG7, in order to have HSP, you need to have 2 mutations in SPG7 in each copy of the gene. What we discovered here is that you can have 1 mutation in SPG7 and 1 mutation in another gene, AFG3L2. So these genes, biologically, the proteins encoded by these genes, they create a complex. They directly interact with each other and they work with each other. And we discovered that people that have 1 mutation in SPG7 and 1 mutation in AFG3L2 can also have motor neuron diseases, including HSP, but we also found several ALS patients and some patients with ataxia. So the concept of having 1 gene and 1 disease is important, and that's most of HSP patients are like that, but there could be HSP patients that have their disease because of a combination of mutations, and it could even be more than 2 genes. Which makes it a lot more difficult to discover. And that could be part of the reason why we still have 35% or 40% in different cohorts of the patients that are still genetically undiagnosed. Now I want to tell you about some current studies that we are doing. So this was what we've already done and we're publishing. Additional things that we're doing is, again, addressing all those that are genetically diagnosed. We are doing a combination of whole genome sequencing and RNA sequencing to find mutations that may be not inside the genes, but maybe a little bit outside genes, but still can have a major effect on gene function and structure. So we are doing a relatively large study on that using these more advanced technologies. We also have a study where we were able to get brains from 4 HSP patients, 4 controls, and 4 PLS patients where we do single cell analysis. The purpose of this single cell analysis means that we can, in each cell from that brain, and you have the different parts of the brains that we're investigating here. So we're not looking at the whole brain, we're looking at specific areas in the brain that are relevant for HSP. And in these areas, we look at each single cell and we look at the genetic makeup and the expression of the genes in each cell specifically. And then we combine all this data. So from each individual that we analyze, from each one of the 4 patients, every patient provides us with millions of points of data in terms of cells with different expression of genes. And basically what we're trying to understand is a little bit more about the neurons that are being affected, which type of neurons, if we can characterize them in a very specific way, are there specific proteins that are important in these neurons, and this could be targets for therapeutics development, etc. So we are quite advanced in this. I think we will have our results in the next couple of months, and hopefully there will be something interesting. I still don't know, but I hope that there will be something interesting that will increase our knowledge and allow us to move forward with some follow-up studies. Sorry. I don't know why the slides are just running forward for no good reason. So the last part of my talk now would be about a couple of therapeutic efforts that we are involved in. So first, I want to let you know a little bit about the infrastructure that we have here at the Neuro that allows us to do that. I'm not going to talk about all the infrastructure. Some of it, Linda, for example, will talk about our biorepository and how it helps us in all of that. So I'm not going to touch that. But what I'm going to touch is how we as an institution help researchers and clinicians move from their discovery in the lab all the way to a clinical trial. So to do that, we have what we call the FIT pipeline, from innovation to therapy, which is a standardized pipeline. We have people working here at the Institute that's the specialty, to bring innovation from the lab all the way to a clinical trial. So from the point somebody at the Neuro discovers a new target, or it has a new molecule or a new therapeutic idea, we help them to go through all these steps, testing that idea in different models, looking at safety, looking at toxicology, all the way to clinical trial development. There's a lot of things involved in this, regulatory things that needs to be taken into account, developing the protocol, things that are more on the administrative side maybe, but clinicians and researchers don't know how to do them. So we provide them with the help all the way to a clinical trial. This is something we started relatively recently, only in the last few years. And this year we are having our first clinical trial like that on a very rare disease in children, Schinzel-Gideon syndrome. There's only 10 children around the world with this disease. One of our researchers developed an RNA therapy for that. And the first child is supposed to get treated by the end of this year. And we follow these steps and this pathway for other therapeutics. And now I'm going to tell you about the case of, again, it's jumping, the case of Gourmand that you might have heard about. I know it made the news here in Canada. I don't know how much it reached the US. Gourmand is a child with a SPAS mutation R499H. It's the same mutation you probably know, the Lillian Blair Foundation. So it's the same mutation that Lillian Blair has and the other kids. It's a de novo mutation. It causes one of the most severe forms of HSP. Usually progression is quite fast, and with very, very severe disability at childhood already or teenagehood. And this child here, Gourmand, the way it all started is his parents contacted me right after he was diagnosed. So he was diagnosed, like I'm sure you've heard it before, diagnosed with cerebral palsy in the beginning. And only at the age of 3 was diagnosed with HSP after he went through genetic testing. So it took them 3 years. The health system in Canada took 3 years to offer this to a child, which is, in my opinion, is horrible. one of the things that we should advocate for is doing this at the moment as the symptoms. So his parents contacted me and asked if I can help. I said I can try. And we moved forward from there, and they asked me, well, what they need to do. And I told them the sad reality that what they can do, the best thing they can do in order to help themselves is start fundraising. Because without funding, we are very limited with what we can do. That's the reality here, at least. And they come from the Punjabi community in the west of Canada, in BC, which is an amazing community, very supportive, and they were able to raise a very significant amount in a very short time, which allowed us to start with this program. So basically, I'm not going to tell you about what motor neuron disease and all of that. You know all of that, but just to tell you a bit more about Gumo, he's 3 years old, 3.5 almost. Currently, he's still walking, but tiptoeing. But it's progressing quite fast, and is expected to lose his ability to walk within a few years, lose his ability to talk. Cognitive impairment will that will increase with time, etc. So it's a bit more aggressive form of the disease, or a lot more aggressive form than others. So what we are planning to do for GUMO is gene editing, which is it's an innovative, novel technology. Only a handful of people have been treated with gene therapy in the world. Only one has been published so far in Philadelphia, a child with a metabolic disease, very different than HSP. And the idea is simply that we change the DNA of the individual. So we're not trying to provide a gene like gene therapy. We don't give small molecules. We try to change the problem at the source, change the actual mutation back to normal. Now, this is very challenging for many reasons. First, it's a novel technology. As I said, only a handful of people received it. But in my mind, in my opinion, this is one of the future of therapies for rare diseases, especially. Once we diagnose them, once we know what the genetic defect is, we can then immediately go into manufacturing and testing these what we call editors that can edit the DNA, can change the DNA. And that's what we're planning for him. We're working with the same group that did the first treatment of baby KJ in Philadelphia from Berkeley. They are developing the editor for us. We still don't know if it's going to work. So in about 4 or 5 weeks from now, we will get the first indications if it's actually can change the mutation, because it is possible that it will not be the case. And then we'll have to think about alternative ways to do that. We have already identified some alternative ways, but I'm talking to you about this because this is currently our strategy. So we're building this editor that the purpose is to change the DNA of the child. And as I said, it's very challenging. We have to reach the brain, which is a huge challenge by itself. So how do we deliver this editor? How, in the sense of where do we put it in the body? Do we put it doing a lumbar puncture in the spine, or directly into the brain? These are questions that we still don't know the answer to. And also, how do we deliver, in the sense of, is it going to be delivered in a viral vector, which is one way that it can be done, or other ways could be what is called lipid nanoparticles. These are like fatty envelopes that can go into cell and deliver the mechanism that does this repair of the DNA. So as you can tell, we are at very early stages, and we're going to test all the different options. And based on these tests, we'll decide what is the best strategy to move forward with. So currently, what we're doing, we are in the stages of design and testing the very early product. As I said, we'll know in 4 or 5 weeks. We'll know if there's indication that it can even work, and if there will be no indication, we will find an alternative way to do it. Maybe something that is called prime editing. Maybe it will be RNA editing. Could be other approaches that we will consider. So we are at that very early stage, and if things go well, then after we identify the editor and identify delivery methods, we will start testing it on models from this patient and from other patient with the same mutation. Now, it's important to understand that this therapy, it's only for this very specific mutation. It will not work on other mutations in SPG4 in SPAS. So if we want to develop treatments like that for HSP in general, it's going to be mutation specific. So we're not even talking about gene specific like gene therapy. We're talking about mutation specific. So that's one therapeutic project we're working on. The second one is also very early stages around calpain-1. So calpain-1 is a gene we discovered, and about the same time, a couple of months after us, back in 2016, another group from the US led by Michel Baudry that you see here also reported that mutations in this gene called spastic ataxia, HSP or ataxia. And since then, numerous groups around the world have reported this patients with this mutation and with mutations in this gene. And currently, we created a database of over 140 patients from around the world with mutations in calpain-1. So it's not super common, but it's not super rare also in HSP. And our plan in this case is to try to develop gene therapy. There's the group from Brazil that I think you might know that is doing the same thing. So we are in discussion with them, and we're definitely not going to duplicate efforts because that would be a waste of time. So we're going to try. We have a meeting with them next week actually to see what they're doing and decide together what would be the best way moving forward. We are very happy. So when I say we, Michel Baudry and I, we combined forces in order to do this. So, we started as separate discoveries 10 years ago in two different labs, but now we're working as one team. And we already applied for NIH funding, and in September, we're applying for CIHR funding to develop gene therapy. We're also looking at, there are some specific small molecules like calpain-2 inhibitors. Calpain-2 inhibitors can increase calpain-1 activity. So we're looking at this direction as well. So in this case, it will not be a mutation-specific kind of treatment. It will be potentially beneficial for anyone with calpain-1 mutations. And I just want to say that all of this started thanks to a patient, again, that contacted us just to show the power that individuals can have. Omar Al-Noemi is from Chicago. He was diagnosed with calpain-1 mutations, and he started this small research foundation, which is just himself, I believe, at this moment, trying to bring more people with mutations in this gene, and is been extremely helpful for us. First of all, he brought us together, me and Michel Baudry, and now the group from Brazil as well. And he is very helpful in terms of connecting and contacting other clinicians around the world that have patients with mutations in this gene. So huge thank you to him. And I'm sure you will hear in the coming years about our efforts in that direction as well. So what we're planning to do, generally speaking, a natural history study, just to understand how we don't have good understanding how SPG76 or calpain-1 HSP progresses. It's like many other forms of HSP. It's very heterogeneous, very different from one person to another. But we still want to try to understand if there are characteristics that are typical or that are unique for in individuals with these mutations. And this will also allow us, we're thinking about a future clinical trial, it will allow us to decide what we want to look at, what outcome we want to test in that clinical trial. In terms of the preclinical research, we have human-derived models from patients. Michel Baudry, he created a mouse model, a knockout mouse model, and we're going to test small molecules, as I said, for calpain-2 inhibitors and also gene therapy. We might also look into gene editing, again, looking editing of specific mutations, but that's only if other approaches we will realize they're not useful. The long-term goal of what we're doing and what we want to do is here in Canada, is to create we want to have a national facility similar to what's being done in Berkeley, but actually a little bit bigger, for rapid development of precision therapy for rare diseases, but also for common later on, because we have some diseases like Parkinson's and Alzheimer's and other neurological diseases that have mutations in genes that could be actionable. And this platform or this facility will be dedicated for rapid generation of gene editing and gene therapy products, so that researchers and clinicians can test them relatively fast. In an ideal world, in the far future from now, probably it will take decades, a person is diagnosed, we find the mutation, immediately we build a gene editor to edit this mutation back to normal. We run the tests on models that are already established and go into clinical trial much faster than what would happen now. Now it's a process that would take years, could take 2-3 years if everything goes well. That's the fast pace would be 2-3 years, could be even longer than that. In the future, it should be taken down to under a year from the diagnosis until a clinical trial. That's at least our goal. So with this, I want to end. I want to thank this is my research team, my own lab, and many of them are not involved in HSP, but some of them are. So they are the ones who are actually doing the job. I'm just supervising them. So I want to thank all of them and thank you for listening. And I know Linda is going to speak next, and after that, I'm happy to answer any questions.
Thank you so very much. I'm sure there's going to be some questions because I know I've got some on my list. But Linda, go ahead. Let's introduce you. I don't think you really need an introduction, but please go ahead and let you take over.
All right. Well, first off, I want people to understand I'm a patient with hereditary spastic paraplegia. My clinical team of doctors is in Ottawa, where I live, but I'm the patient partner of the neuro in Montreal. So I'm retired, so I don't work like salaried work, but it's a patient partner with the neuro in Montreal. All right. I know that's a whole lot of information to unpack. On July 25th, Dr. GAN-OR is going to be doing a very similar presentation at one of my support group meetings. It's on a Saturday, hence it's called the Saturday Social. If anyone wants to join, that would be absolutely beautiful. You can come. This presentation will be on the SPF YouTube channel, which is great. But if you don't have all your questions answered today, you can try again on July 25th if you want. What I'll do is I'll work with Norma, get the link to her, or she'll guide me on how to get the link to everyone that's on the call today. Okay? Just because this is a lot to unpack. Okay. So I am going to start off by thanking Norma for organizing all of this. It's wonderful. So I'm going to talk about CBIG, about the Open Biobank, CBIG, because this is in part how CAN-HSP that Dr. GAN-OR just talked about. It's in part how it gets fueled with information starting from patients. I want to express how very dynamic the platform, the CBIG Open Biobank platform is. It's not one project, it's not one clinical trial, it's a whole bunch of different things. It is mandated by Open Science, which is exactly how we can align CAN-HSP with SP-CERN, with Treat-HSP in Germany and all over Europe. It's how we can align CAN-HSP with other consortia around the globe. Although CAN-HSP is a Canadian initiative, it is absolutely international. The participation is, the whole platform is patient-centered. It's about us. It's the same platform that I built an HSP cohort starting in 2023 with. It's the same platform that I used to add to the PLS cohort back in 2024. And in 2025, I used the same platform along with an ataxia patient out in Calgary. I'm building a cohort of ataxia patients, all using the CBIG platform. It's the platform that feeds CAN-HSP that we just heard about. It's the same platform that feeds Capture ALS. It feeds into Dr. GAN-OR. He's leading an initiative. It's a precision medicine initiative called Energy, NRGI, Energy. It's precision medicine. It feeds, CBIG feeds into that. It's an incredibly dynamic platform. And that's how participation, that's how we fuel CAN-HSP. Okay. So I'm going to, CBIG is a patient-centered platform, right? So it's very, very, very easy to participate. It's meant to be easy, all right? There's 3 steps. It's registration, consent, clinical information, the donation of your clinical information. All that stuff starts with an email to me, lindajlafontaine@gmail.com. That's all it just starts with an email. So for the purpose of this SPF talks, I'm going to unpack all of those 3 steps. The registration, send me an email. I send you a registration link. The registration link asks basic information like your name and your address and all that jazz. At the end of the form, when you click the button submit, it goes directly to the hospital. It does not come to me. All of your information is de-identified, including to me. Okay, I don't get it. The second step, consent. That is permission plus. Okay, it's putting the patient in the driver's seat. It's telling researchers what you will and what you will not do. It's researchers telling you how they manipulate your information, how they'll store your information. It's putting you in the driver's seat. That's step 2. Okay. Step 3, the clinical information, the donation of your clinical information. Now that's all dependent on your consent, but that's step 3. Okay, that's a saliva sample or or an existing brain MRI scan. Okay, now remember in the first step, the registration, you give your address. Well, then CBIG, depending on your consent, sends you a saliva sample. Basically, just empty test tube that you spit in, mail it back. That's it. That's it. That's all. Mail it back, and they extract your DNA from the saliva that they receive. The DNA and the combined DNA and the MRI scans, that fuels a whole bunch of research. That's the stuff that fuels CAN-HSP. It's the stuff that fuels the energy. It's the stuff that fuels. Remember in 2024, Dr. Lapiana came on. She did an SPF talks about white matter rounds. Yep. Fuels that stuff too. Not only stuff in Montreal, all this stuff globally. So it fuels SB-CERN. It fuels IAXON in Brazil. It fuels everything. Spin your globe. That it applies. That's the whole point. Collaboration. Doesn't make sense to redo it all many times over. So just do it once. All that stuff starts with an email to me. That's it. That's all. Oh, and I saw that Hank put my email in the chat. So it's already there. LindaJLafontaine@gmail.com. We're good?
That's very good, Linda. Thank you so very much. I
All right.
really, I really appreciate that.
No problem. And
So,
I will work with you. I will send you the link for the Saturday social.
yeah, please do that, and we can put that on our calendar of events as well, and then that
Yeah,
gives us another opportunity to ask questions of Dr. Ganorth. That would
exactly.
be wonderful. Well, as
Exactly.
you guys see, there's a lot of information that's covered. A lot of thoughts have gone into the work of discovering what's going on with HSP, and it is complex, and PLS, and all the other neurodegenerative diseases, whatever they may be named or however they may have mutated. There's a lot of findings that is absolutely for certain. There are some questions over in the chat box. I don't know if Dr. Ziv, if you could see those, and if you want to just look at those and grab those, that's fine. Or if you want me to read them, I'll be glad to do that. And then if anybody has any questions, if you will, get in line and we'll ask a few questions and then go back and forth. So, let's see.
Yeah, I'm happy to start with the questions in the chat and then
Okay. All right.
answer additional questions. So I'll start just according to the order. So I see Rebecca says she has HSP7 with one parent and has all the signs of HSP. So I think what Rebecca means that she has just one mutation in SPG7. Is this correct, Rebecca? Yes, she's nodding. So it could be another case of diogenic inheritance or a mutation in another mutation. Sometimes they go undetected in SPG7, so it happens. Well, Kristen didn't ask anything, Kristen, but have you been in touch with Omar? And if not, it would be great if you would. So feel free to write me, and I will connect you if you're interested. So I will put my email in the chat as well, so you'll have it. Everyone can have it.
Yeah, I think we had the pleasure of meeting Omar at the annual conference. I think he attended.
Yes, he was wearing an SPG76 shirt,
Yes,
and
yes.
yeah.
Yeah, I
That's
think we have a number of folks that maybe even in our database that have
yeah.
SPG 76 that we might need to look at. And then you got Marina over here that says her daughter has the same variant, same mutation as Gamul.
Yeah. So
So,
the question here from Marina is about that. There's uh,
yeah.
about the base editing, and she said she is asking how long before an inhuman clinical trial realistically could start. And then there's another question. I'll address them separately. So the timing is very difficult to know in advance, obviously. So when I'm speaking to funders or to politicians or to whoever can help, I'm trying to give the best case scenario, and I'm telling them that's the best case scenario. I'm not because I want them to know. And the really, really best case scenario is somewhere between 1 year to 2 years from now. But this is assuming that everything goes by the plan, everything goes well, the editor works, we find the method of delivery very fast that works well, etc., etc. And in science, it's actually quite rare or very rare that everything works. Very often we have to repeat, redo, we fail, we try again. So best case scenario, I would say it's around 2 years. Realistically, it would be probably more than that. It could be between 2 to 3, 3 to 4. It really depends, and it's hard to answer. The second part of the question, how old would the person have to be for it to work? That's also a question that we don't have answer. It's less about how old. It's not about age. It's more about how functional this person still is, and if there's still anything, if the disease is still progressive, and we can maybe rescue some of the neurons and make sure that the neurons that are still alive now are not going to degenerate, to die. So it's important to emphasize that this is not likely to cause any restoration of function. It's not going to reverse the disease. Maybe there will be, assuming it will work, maybe it will create some impression of initial reversal, because if there are neurons that are not dead yet, they're in the process of, and we are able to rescue them. So maybe some some abilities will come back, and it could look like some restoration of function, but it's not going to reverse the process. Most likely, if it's successful, it's either going to slow down, or best case scenarios completely stop the progression of the disease where it's at. So in theory, anyone at any age could go into a trial like that. We're not going to limit age-wise who can go into the trial. It will be more about how much there is still potentially to rescue in terms of the disease progress. So, and these are questions that I don't have the answers to at this point, because we are very far away from designing the trial and really establishing. We'll have inclusion and exclusion criteria like any trial. We will try to make them as inclusive as possible, but there's always have to be some exclusion criteria. So we will not limit by age. Then there's a question from Stephen and Tara about the loss of function and gain of function. Will the edits address both issues? So in theory, in our hypothesis is that yes, because you're fixing the problem at the source, you're fixing the DNA that creates the protein. So moving forward, once you change the DNA back to normal, only normal protein will be created. The toxic protein, the gain of function protein that is being cleared with time, it's not stuck there forever. So we hypothesize that it will be cleared with time, and only normal protein will be created. That's what we aim for. So hopefully it will address both. That's one of the challenges of gene therapy in the context of SPG4, that you need to both provide a functioning gene therapy as opposed to gene editing. What we're doing is gene editing. Gene therapy is providing another copy of the gene, a normal copy. So you provide a normal copies that maybe addresses the loss of function, but you still need to address the gain of function, the toxic gain of function that's been created by the mutation that the individual has. And then you have to remove it using different methodologies. So whoever is developing gene therapy, they're trying to take the approach of remove and replace. So they need to remove the toxic protein. So maybe 2 therapeutics rather than 1 in the same time. There's a question by Rebecca about the contact person for Berkeley. That's Dr. Fyodor Urnov, who is leading the lab that creates the base editors. It's an institution led by Dr. Dunda. She got the Nobel Prize for discovering CRISPR.
Is that Berkeley, California, Dr.
That's Berkeley, California, correct.
Okay, interesting.
Yes.
Good question.
Alexander asking about, is ASO representing a therapeutic strategy relevant for SPG4? He heard from Nicholas Levy, was presenting the N-of-1 collaboration initiative during the I don't know what the GAN-OR summit is. I'm happy to hear about it. In France, that some previous results for ataxia-telangiectasia demonstrate efficiency. So I don't think that ASO alone can be, it could be the removal strategy I talked about removing and replacing. So what ASO does, it's an RNA molecule, and usually what it does, it removes a dysfunctional protein, or it changes it in a way, or it makes it more functional. There are very few therapeutics in the field of neurology in which ASO is beneficial, but I have no doubt that it will be beneficial for many diseases. For SPG4 specifically, I am not sure if ASO alone can be a good therapeutic strategy. It should be accompanied with something else to replace the mutated gene. Then there's a question from Beverly. I guess it is not specifically about what I talked about, but if you are tested positive for ATL1 variant, SPG3A, and hereditary sensory neuropathy, or one or the other, Beverly, it depends. Some people have both, some people have just one. It's very heterogeneous and different from one person to another. Barbara is asking what is motivating me to take on SPG76, which is very rare, and how we can get researchers to have an appetite to do more research on other rare variants or other rare genes, I guess. So first of all, I discovered this gene when I did my postdoc. So I have an emotional connection to it, I would say. But what really drive me is Omar, who contacted me and helped me to connect with others and start this collaboration. So it shows that patient advocates, patients have the power to move things. Of course, everything we're doing now is just in preparation. It's a preparation for therapeutic development. We will need to get funding. So I hope we will. We're applying, as I said, NIH we already applied to, and CIHR, which is the Canadian equivalence of NIH. I'm applying in September, and hopefully we'll get some funding to do that. So the motivation is really, in my case, more because I'm already sorry, I already worked on this gene since my postdoc 13 years ago, and my interactions with Omar. So to answer the part of the question, how do you get researchers to look at other genes? That's a very good question. It's very difficult to answer because there's not a single person. I cannot do 100 projects on the 100 different genes of HSP, and just like HSP, as you know, there are thousands of other rare diseases. So the resources in this case, which are the researchers, are limited. Not to mention, again, I spoke about it too much maybe in this call, but funding, also a resource that is very difficult to get. So by advocacy, by contacting researchers, that would be the way to go.
Well, let me throw this at you, Dr. Ziv, and I really appreciate the comments and the questions that everybody has brought up. One of the things that you talked about, much of the work that you described is mutation specific, like gene editing approach for the SPAS, the R49OH. Given the complexity and the diversity of HSP genetics, how do you see that opportunities for therapies that target shared biological pathways or cellular mechanisms across multiple HSP genes, rather than just one mutation at a time?
Yeah.
What kind of data, what kind of models, what do we need to make those kind of pathways realistic? I mean, that would get a lot of other people involved in this.
Yeah, I think that's a very important approach that we should not abandon. Working on molecules or therapeutics that can affect a pathway that includes multiple HSP genes and potentially help all the carriers of all mutations in this group of genes. So this is something that should definitely be done. I think it's very important. I decided to go towards this direction, which I know is a lot more limited in the amount of people it can help. At least in the first pass, we're trying this mutation. After that, we'll try another one and another one. But the hope is, as I said, is to increase our capacity not to do this for one individual, one mutation at a time, but do it to 10 or 20 or even more in the future. That's what we hope for. And the reason I'm going for this specifically is because from my perspective as a geneticist, this would be the ultimate solution for a therapeutic solution for someone with HSP or with many other rare diseases, where you correct the problem at the source. When you give a drug this correction can be done just once. It's a one-time treatment. Maybe you need to do it again, twice, although that has many other challenges because you cannot do it with a viral vector, at least not at this point. So it's very, for me, it's very appealing, this idea that we can completely correct the problem and fix it for the patient forever. The problem with my approach is exactly what you said, that it will only benefit very small amount of people at a time. I hope that we'll be able to build the capacity to do this at large scale and not just for one patient, one mutation at a time. And that I'm sure it will happen. The question if it will happen in 5, 10 years from now, that we can do that, or if it will happen in 50, 100 years from now. But this is where we're going towards very precise therapies that change that correct the problems at the source.
Well, and as you can see, once you have a conversation about a gene mutation, you actually find out that there are other patients, across the world that also share in that. And that may become even more important is for folks to understand their real genetic history, their information about themselves.
Yeah. And just to say that this specific mutation, for example, if you calculate how many people around the world have this mutation, based on the statistics, we know about 5 out of 100,000 people have HSP. And we know that out of those, about maybe 20 to 40% have SPG4. And out of those, maybe 1% or 2% have this specific mutation. We still reach a number of about between 1,000 to 2,000 people around the world that have this mutation. So it's not a negligible number, right? Many of them, they have no idea that they have this mutation because genetic testing is not available in large parts of the world. But statistically, these people are out there. So that's another thing
Really
to
what
remember.
needs to resonate is that everybody needs to understand what is their information, what
Yeah.
are their gene mutations.
Yeah.
We've heard so much about SPG numbers, but we're not only dealing with particular SPG numbers, you're actually dealing with a gene mutation within that SPG number,
Yeah.
like a subset.
But another thing to remember is that our experience here will greatly, improve our ability to do this more and more. This is the first one we're trying. It's in a way opportunistic because the funding is there and the motivation is there and everything came together at one time. But the experience that we're going to get from this and the skills and the technology, they will be available for everyone else later, so, which will make the process of developing additional gene editors easier and faster.
And when you mentioned, Dr. ZIV, that you had a grant at the NIH, is that the NIH in the United States?
Yes, there's only one NIH that I'm aware of. Yes, so Michel Baudry is also in California. So he submitted this grant to the NIH
Okay.
in the US. It's an R21 grant.
Okay.
So we should get the results in November. We will know in November if we got it.
That would be fantastic. And you mentioned funding a number of times, right?
Yeah.
I think maybe like 8 or 10. So
Yeah, too much.
well, but it's important. I mean, that is, we've understood a while now that, it's kind of a pay to play. I mean, it's kind of, you got to have the money in order to match or have that available for the research and the science and the labs. We get that. What do you see? What is that amount? What is that amount and how long does that take you for the work?
Amount for what? For
Yeah, funding. When you say you need funding, what do you need funding for? How much funding do you need? How much money?
Yeah, so I'll give you the example of Gourmand's family. So in order for us to develop something from zero all the way to a clinical trial and give it to an individual, now our estimation in Canada is between $8 to $10 million.
Okay, 8 to 10 million.
8 to 10 million, yes.
Okay.
That include all the different stages of development. So there's the very preclinical work that we're doing now, testing on different cells, animal models, mice. We'll also test it on marmoset, on primates. And then what costs the most money is actually the toxicology part that is being done by companies that are experts in this. Usually the cost of that alone is between $2 to $4 million. that part of alone of the toxicology. And then there are the costs. If everything goes well, there are the costs of the clinical trial itself. So our estimation is between $8 to $10 million. So it's a substantial amount. But we also foresee that these amounts will drop significantly. For example, if we do have a facility that can manufacture these things at scale, and if we can, instead of testing, now we're testing one editor, like one mutation at a time. If we test 10 at a time or 20 at a time, we already have the instruments to do it. We have here at McGill, we have a huge robot that can test RNA therapies on multiple cells and using multiple concentrations, multiple delivery methods, et cetera, et cetera. We're going to use like a small portion of it to just test that, but we do it at scale. We do it for 100 different mutations at the same time. It will save a lot of money. So I predict that the costs will drop down significantly in the future, but they're still going to probably stay in the millions. But maybe instead of 8 to 10 million, it will be 2 to 3 million. So that's my estimation.
Well, I appreciate that because, really at the end of the day, that's what it comes back to is we've got to figure out financially, where do we invest financially? How do we get to the next step?
Yes.
It's got to come into play. And, obviously NIH and these other organizations, they have the big bucks, they have the big dollars, and they can start looking at these types of neurodegenerative diseases and how do we fix some of the population. That would be wonderful.
Agreed.
Agreed. Well, I love that. Okay, anybody have any questions? We are over time, and I do apologize for that, for all of you that have stayed with us. We'll entertain one more question. If anybody has got that, raise your hand or throw it over into the chat. And you're going to have another opportunity, as Linda had mentioned. Put this on your calendar on the 25th. Linda does a talk show as well, and she's going to have Dr. ZIV back on, and then we can find out some more information and be ready for some more questions. So you're not caught off guard. So get yourselves ready.
Yes, July 25th, 2 o'clock. That's Eastern, so that would be 1 o'clock for Central.
Okay, 1 o'clock Central. I'll put that on my calendar. See what I'm doing for that day.
Yeah.
Let me try to join you. I love learning. I love finding out what's going on in this industry and the things that are happening, because it's absolutely remarkable. All of the work that is going on everywhere. And if we can take what we know in this place and in this place and pull all of these together like a wheel, like a spoke in a wheel, right? Everybody's got a piece, everybody's learned, everybody knows what's happening. And we've got to learn from everyone to try to get to some type of therapy, some type of therapeutic, something that can make a difference and get us to a treatment. Everybody with me on that?
Oh, yeah.
Yeah? Pastime, right? Pastime.
Yeah.
Well, there's a lot of takeaways, and I know I have tons of more questions that I want to come back, Dr. ZIV, and talk with you again about, but I really appreciate your being here today and talking with us, and we're going to need to stay in contact.
Yeah, and I'm happy to come back again if you'd like me to.
I would like for you to.
Okay.
All right, you guys. Thank you so
Goodbye.
much, everybody,
Take
for
care,
joining
everyone.
us.